guys, in this video I'm going to be showing you a super cool experiment on how to extract the DNA of a banana. So DNA or deoxyribonucleic acid is a long molecule that contains a unique genetic code. Like a recipe, it holds the instructions for making all the proteins in our body. It is made of a chemical called deoxyribonucleic acid or DNA for short. So let's not waste any more time and get started with the experiment. You need not think that this is very complicated. This experiment is super easy. You can make this experiment successful with only few things that are available at all of your homes or shops near you. So the things that are needed to make this experiment are some table salt, some slices of banana, half a cup of hot and cold water, a filter, a gauze, some liquid soap, a few glasses, a bowl, a spoon, and some isopropyl alcohol that is available in sanitizers. So the first step is to make an agent. To make the agent, take the glass and add some salt. Liquid soap, and add half a glass of steaming hot water. Just please use really hot water for this experiment. After adding the hot water, take the spoon and mix the agent well. Mix it thoroughly because the salt should completely dissolve in the hot water. Then, after mixing it well, keep it aside. The second step is to take another glass and add a slice of banana to it. Use one whole banana. After adding the bananas, add half a glass of plain water. After adding the plain water, add 9 to 10 tablespoons of the agent you have created before. After adding 9 to 10 tablespoons of agent, mix this banana, normal water, and the agent. Here is the mixture after it's blended. The third step is to keep a bowl and place the filter on top of it. Then take the gauze, which is a thin netted sheet, and cut it into 3 to 4 thin strips. Cut the gauze into thin strips. Now I'm going to place it on top of the filter. Placing the gauze in the filter, take the mixture you have blended and pour it on top of the gauze. It will be semi-solid and semi-liquid, but don't worry about it, you're gonna filter it. After adding the mixture on top of the gauze, take a spoon and mix it well. This speeds up the filtering process so you don't have to wait for more than 15 minutes. You need not worry about the gauze because it doesn't tear like tissue paper. After 15 minutes, you have to take the filter out, and when I take it, the filtrate is collected in the bowl. You can see the filtrate that's collected from the mixture after filtering it. The fourth step is to take another glass and pour the filtrate that you have filtered before into it. I'm gonna set aside all other things except the filtrate and the isopropyl alcohol so that you can get a clear view of the experiment. Use cold isopropyl alcohol, guys. Don't use hot or normal one. Use cold one. So the fifth and the last step of this experiment is to add the cold isopropyl alcohol into the filtrate. After adding it, wait for 5 minutes. After 5 minutes, you can see a little amount of change in the filtrate. You can see white spots here and there. Now I'm going to lift and you're gonna see what's inside. Can you believe it, guys? We've just extracted the DNA of banana. The thing I'm lifting is the DNA of banana. This is a closer look. So we've successfully extracted the DNA of the banana using this method. You can also extract the DNA of strawberries and peaches using the same method I've used in this experiment to extract the DNA of the banana. So this happened because first, when we added the agent, it destroyed the banana cells and the DNA dissolved in water. Next, the DNA precipitated in the isopropyl alcohol and we collected it. This is the explanation. So I hope you guys enjoyed my video and learned something from it. If you did, please subscribe our channel, like, share and comment my video. I'll meet you with yet another cool experiment. Until then, bye guys!